So I would like to tell you about our ongoing efforts to understand the origin of folded proteins. I reported on this at previous 3 SIG meetings in Berlin and in Dublin, and I'm really grateful to have the opportunity to tell us where we stand today. In essence, nature relies on folded proteins, on the catalytic activity of folded proteins for pretty much every process ongoing. And the folding of proteins is a difficult property. Nucleic acids fold robustly. Witness the PCR reaction, where you go through repeated cycles of melting and annealing, and you keep getting your template back. With proteins, that doesn't really work. So if you toss an egg into a hot pan, you get an omelet. And when you cool it down, you have a cold omelet. And in nature, things run in very similar ways. Once proteins are denatured, they have to be degraded and resynthesized. Now, part of the reason for this is because folding is a very rare property. Most random polypeptide chains that you could synthesize will not have a folded structure. And from large-scale screening efforts, Hecht, Plückthun, Shostak, Winter, we know that the folding yield of these libraries, even though in most cases they were biased libraries, was less than 1 in a billion, and quite possibly much less than that. Now, in modern proteins, nature has bypassed this problem by assembling new folding proteins from domains, from pre-optimized folding entities which are being recombined and differentiated in order to produce new properties. However, the question that we're trying to understand is, where do domains themselves come from? They're much too complex to have arisen by abiotic properties. And so the hypothesis we started on 15 years ago is that the first folded proteins arose from an ancient set of peptides which themselves evolved in the context of RNA reproduction and catalysis. And these peptides were fully dependent on the RNA scaffold for their activity and their structural properties. However, by being optimized for specificity, and thus to exclude water with the scaffold, they, at some point of complexity, found that they could exclude water between each other and become independent of the RNA scaffold. So in this view, folding would have been an emergent property of complexity arising from progressive cycles of peptide RNA coevolution. And down here you see a number of folds of today which can be traced back to fragments which might very well have been used in either in recombination or in repetition to produce folding. So when I started out on this, I was very strongly influenced by my boss. I was at SmithKline Beecham, and my boss was David Searles. And that may ring a bell with quite a few of you. But David was very interested in the informatics of language. And so every time I had an idea, he would ask me, well, how does that compare to the Chomsky hierarchy? And in fact, we had very many conversations. And in the end, much of the basis of what I'm going to tell you today was developed in these conversations. Now, he pointed out to me the tremendous evolutionary potential of language and our ability to trace evolutionary events. And so let me give you one example. Let's start with this Semitic root, kane, which means reed, and which gave orthologous descendants in all Semitic languages, also in Hebrew and Arabic. It came by lateral transfer into Greek and Latin, and then via the French can into English as cane. So we have a 5,000-year chain of orthologous descent denoting a swamp plant. Now, in ancient times, there were several instances of paralogy, the Greek used the straight and rigid property of reeds to make rulers, measuring rods, and they called them kanon, from whence comes the canon as the church rule and the canon as a musical form sung by rules. The Greek also used the flexible property of young reeds to weave wicker baskets, which they called kanastron, and from there comes the canister and also the game of canasta. The Romans used the round and hollow property to assemble pipes, water pipes, and they called them canales, from whence comes the canal and the channel and the canyon, and then also things like the can and the cannula and the cannon and the cannelloni. So if we transpose this to the protein world, and believe me, the analogy between language and protein evolution is absolutely tremendous at almost every level of resolution. Here we track a fragment, which we have described. We call it the beta hammerhead. We have tracked it into a ribosomal protein, which is still dependent today, I believe, on the ribosome for folding, but which gave rise to a cytosolic domain, a subunit of the exosome RRP4, which is autonomously folded. In the time of the last common ancestor, presumably before the last common ancestor was reached, a duplication of this fragment gave rise to a new binding pocket capable of binding biotin, and that ancestral fold was the precursor of pretty much all biotin-dependent enzymes of today. And here we have two more items of porology, a subunit of RNA polymerase, which today is not visibly involved in binding the RNA, and a part of the regulatory subunit of thymidine phosphorylase. Actually, it's not a subunit, it's a domain within these. So we see some regulatory activity, some new catalytic activity, and the original RNA binding activity. Now, when we set out, we decided to try to track this evolution, just like linguists did it for languages, to track it in sequence space. Also because sequence space is de facto practically infinite and therefore would retain most probably traces of this descent. And for this, we developed a suite of tools for sequence analysis, the HH suite. In fact, Johannes Röding, who was a postdoc with us at the time, built it, and we deployed it in the MPI Bioinformatics Toolkit, which is substantially built on these programs. And today, Vikram has taken over the toolkit, and you see it has a new look and feel, but it still does pretty much most of the same things. Now, with these tools in hand, we started looking systematically at proteins of the SCOPE database, and we found fragments that were locally similar in globally dissimilar folds. And then we checked the structure of the fragment in all cases to see a confirmation of the prediction that they are the same fragment. So structure was not an input into the search. It was a control mechanism. So the fragment was defined exclusively at the sequence level and then tracked back. And you see that the fragment is actually very well conserved, even though the parent proteins or the originator proteins are quite dissimilar, also in chemical activity. Now, Vikram has done this systematically over SCOPE, and these are 40 fragments that we found where the statistical significance was good enough that we felt we could publish them. We have a few more, but we're working on them. Now, these fragments are greatly enriched in the ribosome. Almost a third of the fragments that I've shown you come from the ribosome, or are present also in the ribosome. And we think that as the last survivor of the RNA world, the ribosome may be an excellent source of documentation of the transition from unstructured material dependent on RNA to fully folded autonomous proteins. And so if we look at this, you see these, you see these purple circles. They mark the peptide ligation center and the decoding center of the large and small subunits respectively. And we're using here the geometric center of the ribosome. And within 50 angstroms of the geometric center, there's practically no folded material. There's little protein material, and it's essentially without regular secondary structure. Now, in a sphere of 70 angstroms, we start seeing quite a bit of secondary structure, which is also more and more frequently arranged into supersecondary structure elements. And at 90 angstroms now, we see quite a few supersecondary structure arising. And in the outermost shell, we see quite a few things that look just like folded domains, but they're still dependent on the RNA. And so we could consider the cytosolic protein as being the fourth shell from this ribosomal representation. And we think that ribosomal proteins essentially allow us to chart this path of progressive emancipation of polypeptides to folded proteins. And so we have analyzed a number of these experimentally, and I would like to show you what we did with this fragment here. This fragment is found, I need 2 fingers, is found here. And we think that we could use it to generate this kind of structure, which is a TPR repeat. Right now, we don't want to propose that this was the parent of this. We think rather that there was a population of such hairpins in the RNA world, and that they gave rise to a lot of different folds, and that this and this arose divergently from this original population of hairpins. Just like this, right? The only repetitive one is the TPR. All the other ones have the hairpin in single copy. And so we took a ribosomal hairpin here from RPS20 and tried to reproduce this here, which is a 3-repeat TPR. You see here the superposition of the yellow hairpin to the blue hairpin. And in essence, Hongbo designed, we were hoping, of course, that something completely unmutated just by amplification would give us a folded structure, but he also used protein design to find mutants that would help us get there. All of these are folded. We can show that. This one is the one for which I will show the crystal structure, but now we also have some evidence that the completely unmutated form, if we amplify it sufficiently, namely 7 or more times, will fold. And so this is the construct. And the design is in gray. The crystal structure is in cyan. And you can see that we did generate a TPR protein just by amplifying a hairpin. Right? The angles are all correct. The registers are correct. This is exactly a TPR repeat. Now, we don't have the stopping helix. I won't go into what that means. Oh, I will go. I had forgotten I had left the slide in. In essence, the structure we built is a homodimer, antiparallel, and the 2 helices here satisfy for each other the position of the stopping domain. So the stopping helix has no place in this protein, and so it's out in solution. So in essence, to conclude, RPS20 is a small ribosomal protein. It's unstructured. In the absence of RNA, we've tested that. The N-terminal hairpin has sequence similarity to the TPR consensus and could have originated from an ancient population of hairpins that also yielded the TPR. And a small number of mutations, quite possibly no mutation, are sufficient to allow this hairpin to produce a folded structure by repetition. Now, we did introduce all the mutations together in Thermus thermophilus into the genome. There's no phenotype. Growth is normal under any condition we could test. So they look to be largely neutral and therefore quite possibly having been explored in the course of evolution. And each of the mutations occurs individually in at least one Thermus species. So it doesn't look like Thermus minds very much the mutation at these positions. Right, now the conclusion of the conclusion is we have tried to recreate here a scenario for the origin of a folded protein from an intrinsically unstructured peptides following evolutionary information which comes from our large-scale analysis of protein domains. Thank you very much.
One question for you is, so you showed that you suggest you already found 40 important peptides that play an important role in protein folding. How many such peptides are there in nature, according to your expectation?
Well, okay, let's say these 40 are the ones where we have good statistical support. I would imagine that the total number of those that we can still see today, because many must have died out at some point, the total number will not really exceed 100, 150. Right. I mean, we can already account for most protein, the very well represented protein superfamilies, we can account for by these fragments. Interesting. Right. So I think it's a bit like the English language, which has, I don't know, 50,000 words, but a few thousand words account for pretty much everything we talk about.
Well, these peptides cover the whole structure space of all the possible peptides that are important for protein folding?
Difficult to say, but I think of the proteins we see, they do cover it. But I think that protein folding space that we see in nature is not the one that's possible. I think much more folded material is possible and has not been explored or has not been retained because it's not useful. Yeah.
Okay,
that's Arne.
Oh, okay. Okay.
Okay.
Okay. Let's thank Andrew for the nice